Hello there and welcome to this Biology Mind A-Level tutorial on protein structures, where we're going to be having a look at primary and secondary structures. So our key learning objectives are number 1, to have a look at the formation of the primary structure of the protein, and number 2, you've guessed it, to have a look at the formation of the secondary structure of the protein. So there are various different types of structures of the protein, from primary to secondary to tertiary to quaternary. So today we'll just focus on these two, the primary and the secondary. So I'm going to give you a couple of moments here to pause the video and have a go at labelling which one is which in terms of structure. So which is the primary structure, which is the secondary structure, which is the tertiary, and which is the quaternary. So pause the video now if you want to have a go yourself. Okay, so we'll talk about the answers now. So this is our primary, because remember our primary is just our chain of amino acids. It's our 2D structure. This is our secondary, which is when hydrogen bonds form between the primary structures. So within this primary structure, various hydrogen bonds form and cause either a alpha helix or a beta pleated sheet to form. We'll go over this in a little bit more detail over the next few slides. And then the tertiary is the interaction of these secondary chains together. And the quaternary is the interaction of these tertiary chains with each other. Again, we'll look at that in more detail in the next tutorial. Actually, we won't cover it in the next few slides. So let's kick off by having a look at these primary structures first of all. So the primary structure of a protein is the sequence of amino acids that makes up its polypeptide chain. So this is a definition that I want you to learn, because this is quite a common exam question to be asked, and it's really easy marks and you can just learn this definition. So it's flat and 2D because it's literally just a chain of amino acids joined together by what? Do you remember the name of the bond that joins amino acids together? We had a look at it in the last tutorial. So it's our peptide bond, which is formed by condensation reactions, remember? So that's our primary structure. So now let's have a look at our secondary structure. So our secondary structure involves hydrogen bonds forming between the R groups of amino acids. So we had this flat amino acid chain, and when hydrogen bonds form between the R groups of these amino acids, we get them bending in different ways. And this forms either the alpha helix, which forms due to coiling, or the beta pleated sheets, which form due to folding. Now, make sure you remember what hydrogen bonds are and how they work. This will just be a little recap that you can just do, and then this will make a little bit more sense as well. But it's really important that you remember that it's hydrogen bonds that are formed within the primary structure to form the secondary structure. Okay, so if we look at the primary structure alone, it just has peptide bonds in it, whereas the secondary structure has the hydrogen bonds on top of that, which form these various different patterns, either the beta pleated sheet or the alpha helix. And this is just another diagrammatic way of showing our secondary structures. So remember, this is our chain of amino acids, this is our chain of amino acids, and the red dots are our hydrogen bonds that have formed between our R groups. All right. Okay, so that completes our tutorial for today. Well done for following through, and in the next one we'll be having a look at tertiary and quaternary structures.